So today for the graduate section, we'll talk about yet another kind of contrast. So this is called magnetization transfer, or MT. So they call this MT contrast. And so that's the great thing about MRI compared to something like an X-ray. Like an X-ray, you basically got an X-ray. You do the X-ray, you can't really change the contrast, but MRI is incredibly flexible, so you can change, the contrast from, you can get T1 contrast, as we've all learned about in the homework, or T2 contrast, or proton density contrast, or perfusion contrast. We'll talk a little bit about that when we talk about time of flight. Or diffusion contrast, what direction is something diffusing in. Or magnetization transfer. So it's yet another kind of contrast that you can set up in a scan in order to essentially completely change the colors in the scan and emphasize different things. So one of the problems that I've mentioned several times, but we could sort of mention it again, is if you look at any one image, the problem is two different tissue types, completely different tissue types, might have the same color. So if you look at a typical brain image, you have some skin like this, and which is relatively brightly colored. And then there's a little layer of fat under it that's brightly colored. Depends on how big you are. If you're really big, that can be really big. Then you see the CSF. Then you see the skull, and the skull is black. It's got there's a little bit of sort of marrow of the skull. Like this is called the outer table of bone and the inner table of bone. So the skull, basically this is the skull right here. That's the skin, and then this is fat, and then the skin, and then you get into the what's inside the head, and what's inside the head is sometimes you can see the little dura. That the dura is like a leathery layer that kind of protects the brain. And then further inside, you've got the actual brain, which is bright again. So here's the gray and the white matter like that, and the white matter's really bright. And then what's in between here? You got the CSF. It's dark, cerebrospinal fluid. So if you look at that, and then you got air out here. So the air is dark. Why is the air dark? Well, it doesn't have any protons in it hardly. But the skull is dark. Why is the skull dark? Well, the skull is dark because the T2 is so short. The T2 of this, it turns out there's a big signal coming out of the skull. So you do an RF pulse and a big signal comes out of the skull. But the problem is the skull has such a short T2 that the signal completely decays away. T2 and T2 star decay away the signal completely, and so by the time you turn on the receiver to receive a signal, it's all gone. So that's another thing, you couldn't record the echo fast enough. There's tricky ways to try to do that, to actually get an echo time that's under a millisecond, so you can actually record the skull, and then suddenly you see there's a big signal there. So that doesn't have any protons. That's black because the signal decayed away. CSF, however, is making a big signal, but it will be dark in a T1 scan because it will have taken longer to recover, and so you'll see a different. So many different things. So that's why you want multiple contrasts, because if you use different kinds of contrast, then you could sort of like pull apart some of these things that are the same color. That would be, for instance, the same color in an X-ray. But you can sort of tweak and sort of fiddle with it. So MRI is tremendously sort of flexible that way. And one of the things that helps it out, being even more flexible, is magnetization transfer. So we'll talk about magnetization transfer, and that involves something called off-resonance excitation, which I talked about before, but is going to be relevant when we talk about perfusion or arterial spin labeling. And so what I'm going to also talk about today is how time of flight scans work, which is sort of a kind of a perfusion type scan that came out a long time ago. And so we'll try to explain how that works. But so as I was watching my video from last year, I said, like, I don't really completely understand. I don't understand magnetization transfer, especially the physics of how it works. But we'll just go over sort of the main points and how we actually use it. And so, what is the distinction? Well, it's the distinction between what we might call sort of bound protons in macromolecules. So you have a lot of proteins in your macromolecules. So you have a lot of proteins and things like that have protons in them, and those protons behave quite differently than the protons, sort of another sort of generic class of free protons, and those would be like free protons in CFS and cerebrospinal fluid or blood or tissue fluids. So there's a lot of fluids around cells. So those things behave quite differently from each other in terms of how they resonate. And so one of the things that is a difference is that the range of resonant frequencies of these bound ones is much greater. So and how can you do that? Well, how can you test that? Well, you can just go into a regular NMR or chemist's magnet, get a bunch of, dissolve a bunch of protein in water, and then scan it and just look at what frequency comes back versus just the water. And you can really, easily see the difference. And the difference is you get this broad band of different frequencies. So if you just do an RF pulse containing all frequencies and then look at what resonant frequencies come back when you record it, you see a big broad peak like that. Whereas if you just do plain water, you'll get this extremely sharp peak like that. A little sharp peak like that. So that's frequency again. So this is the frequency of the signal. So this is freak of signal coming back after we sort of hammered it with a hard pulse that has all different frequencies. So what's the basic plan of magnetization transfer? You can sort of, so if you want to sort of examine these guys, you're only hitting a small number of them when you use a very frequency-specific RF pulse that's designed to excite the water protons, which is basically what are, out of all the protons in your head, most of them are water. And so no matter what, we're going to sort of have to go after the water. But if you want to examine these guys, then you're only going to be getting a small percentage of them if you do a narrow frequency, a narrow frequency band pulse. And so what you can do is set your pulse, off to the side. So if you set your pulse off to the side, so this is our off resonance pulse. It's not off resonance for the macromolecules, it's just off resonance for water. So to put that in quotes, it's not really, it's on resonance for the macromolecules. So if you do that, then what you can do is eventually look at the signal, which is going to be bigger, coming from the water, and look at how that off-resonance pulse, which excited these guys, affected the water signal. And that's the transfer, the magnetization transfer part. So basically the plan is just, an off-resonance, meaning, water resonance, off-resonance RF pulse. And this is kind of like inversion recovery. So like when we talked about inversion recovery, what we said is you can think of it as, or what they would call it in the MRI literature, is a preparation pulse, because it doesn't actually make an image. It just kind of conditions what kind of signal you're going to get out of it. And so you do this you do an inversion pulse, and then you do a 90 and do all the things you need to do to actually make an image. That you can sort of think of that as kind of a whole module of its own. And so you just have this preparation pulse, and this preparation pulse will sort of color the image in a particular way, the whole image. And the off-resonance pulse is like that. It's kind of something that sort of colors the whole image. So you do that pulse, you get a little signal back, but you ignore it. And then you wait for magnetization transfer, for MagTransfer. And the way this works is you're looking for magnetization transfer from the transverse magnetization that you created here to the longitudinal magnetization. So it goes from, MT of macro molecules to ML of water. And what does it do? It actually attenuates. So the result of the transfer, the result of this transfer of magnetization from the transverse magnetization of the macromolecules to the longitudinal magnetization of the water is, result is attenuation. So like, reduction. So attenuation of ML. So it actually darkens. It doesn't matter, darken or lighten. You can flip it around if you want, when you're viewing the image. But that's the result. So what you're trying to do, what is the goal of this? Well, the goal of this is, could we sort of make an image that's somehow weighted by properties of the macromolecules? So different parts of the image that have different compositions of macromolecules will have different amounts of magnetization transfer from the transverse magnetization of the macromolecules to the longitudinal magnetization of the water. And then when you do the image, since you've messed with the starting longitudinal magnetization of the water, you get some contrast later on. So so you can think of it as like a preparation pulse. So what you would do is you would first do this magnetization transfer pulse, MT pulse, and then do, whatever you need to do. Say you were doing a gradient echo where you have like a whole bunch of, alpha pulses to record your image. So you basically just do a magnetization transfer pulse, wait for it to sort of leak into the water protons, then make an image. So here's where we make our image, and that's our whole pulse sequence for making the image, which we haven't talked about the details yet, but a whole bunch of parts of that, image, etc. So there's going to be thousands of things here. So it's quite easy to so if you're on the scanner, if you want to do a magnetization transfer, all you do is just tack in this extra thing before you start, and then the image that you get out afterwards will be magnetization transfer weighted. So attenuation, okay, and then for make image, So one of the things, if you're thinking about, like, say, we just talked about slice selection, one of the problems is this happens all the time, right? It's kind of like, how can you avoid this? So, it happens all the time. And it's one of these things where it's kind of like T1 and T2 weighted. Like, it's always T2 weighted a little bit, and it's always T1 weighted a little bit, and there's always weighted by proton density. You can't avoid it. And it's always magnetization transfer weighted. Because just think about it. Every time you do a hard pulse, so if you're doing a gradient echo image, you might have 100,000 hard pulses. So every time you do a hard pulse, what is that? That's it's a narrow thing in time. And what is that Fourier transform to? That Fourier transforms to like a giant sink, also kind of known as everything. So what is everything? All frequencies. So isn't that an off-resonance pulse? It is. It's on resonance. You're aiming for the water. You're hoping that one of the middle frequency is the water, but it's hitting all frequencies, and so it's doing magnetization transfer. So every time you do, when you're doing the gradient readout, you've got a little bit of magnetization transfer in there too. But what about a frequency selective pulse? Even a frequency selective pulse will cause this. So you'd figure okay, so we don't do a hard pulse because that's a sloppy one and it hits all these different frequencies. Let's just do a pulse that has a frequency range that looks more like what we're trying to excite. So if you do a frequency selective pulse, well, what is the thing you do? Well, you got the person in there, and what do? You turn on a gradient. And what does the gradient do? Well, say it was the Y gradient, it changes the resonant frequency. And then we do, in the Y direction, how does it do that? It changes the Z component as you move in the Y direction. And what are we doing that for? Why are we turning that gradient on? Because we're trying to select a slice like this that has the resonant frequency, just the resonant frequency that the pulse has, so that the pulse will sort of select that slice. Well, what about the guys over here? Well, they're getting magnetization transfer because they're essentially have a gradient that has changed the resonant frequency, and now we're giving a frequency selective pulse, which is an off-resonance pulse. So it turns out you can't you can't avoid it. It's always happening a little bit, and so it's mixed in there, in the image. So what are these, magnetization transfer used for? Well, like I said, why haven't you heard about this? If you know anything about MRI, you probably haven't heard of this. It's partly because it's not that common, and one of the reasons is the signal to noise is less. So the overall signal to noise is less. It's partly because not that much, you don't get that much attenuation when you do the pulse, and doing the pulse, you can't do a perfect pulse, and you hit some of the signal that you're trying to actually record. So there's several reasons. So the signal to noise of a magnetization transfer, and you've got to wait around more. There's more time to wait around. So one of the things is it generally will have a lower signal to noise. So because of that, people don't do it as much. The images look more grainy. One thing you can really use it for is preserved tissue. Now, you might wonder, why do you want to do that? So one of the things with preserved tissue, how do you preserve tissue? You put it in something like formaldehyde or phenol or something like that. And what does that do? Well, formaldehydes are an extremely reactive molecule. And what formaldehyde does is it essentially sticks everything to everything else. It just makes like a giant meshwork of bonds where everything is stuck. And what's the purpose of doing that? Well, you're just trying to avoid other kinds of life eating it. And so when when the bacteria comes in to try to eat this thing, and it's just this giant meshwork of hard bonds that it doesn't have any enzymes to break apart, it says doesn't look like food. And so then the brain will sort of stay preserved. So, but why, since we have methods, if the brain is dead, we could sort of slice it up, section it up, stain it, and look at it that way. Why would you want to scan it instead of just preserving it and sectioning it? And one of the problems with sectioning it, especially a human brain, it's really big. Like, how many sections are there in a brain? The sections are usually like about 1/20 of a millimeter thick. So you'll have like 20 sections at least, per millimeter. So think about that. There's a millimeter, and you can have like 20 slices per millimeter. And each section is really big. Like, it's this giant thing about this big, 1/20 of a millimeter thick. You can imagine that's kind of a difficult thing to deal with. And so you float it in water and try to arrange it onto a slide without being too distorted. And what happens? Well, each different section is slightly off and squished and non-linearly squished. And so if you're trying to sort of make a reconstruction of a brain from sections, it's quite a difficult process. You need a whole bunch of processing to actually sort of unstretch all the slices and to kind of line them up with each other. What about just sticking the thing in the scanner? Yeah, the big thing about a brain, a preserved brain, is it doesn't move. And so I always make the joke, like this next scan is going to be 24 hours. Just try to stay as still as possible. Don't. And for the most part, the brain will follow those instructions, stay completely still, and so we can scan for 24 hours. And if you scan for 24 hours, you can make a higher resolution image because you've got a lot more signal to noise to work with. And so there's a lot of work on preserved tissue. You might even want to do this even if you're intending to section the brain, because it's not distorted. So you can even scan it entirely in the head. It's slightly gross, but you can put a whole head in there, scan it before anything is changed, before you take it out. And then you have like a model of what it's supposed to look like, and then you section it up, and then you could stretch all the distorted sections back into alignment. So I've done some stuff like this with the human cerebellum, for instance, to sort of reconstruct what the cerebellar cortical surface looks like with preserved tissue. And you can scan for hours and days. So that's the reason why you might want to do that. So there's a whole field of sort of scanning dead things. Okay, so that's magnetization transfer. So what are some of the things that are done with this? And so one of the things is to increase the contrast of a time of flight scan. That's time of flight. That's the TOF. And so what's a time of flight scan? So a time of flight scan is kind of it was an early form of what you would call, MRI, MR, or magnetic resonance angiography, to sort of visualize the blood vessels. And there's various ways of trying to visualize blood vessels. Like, if something's gone wrong with your blood vessels, they'll take you in and do a catheterization, where you fish in a catheter into a leg vein, and then fish it all the way up into the heart, come out of the heart, bend around, go someplace else. And then you can continuously turn on an X-ray and inject a little contrast, something that actually will show up very bright on the X-ray, into the, or dark on the X-ray. It's actually dark because it's absorbing the X-ray photons, and then you'll see a dark spot where the arteries are. And what are they looking for? I would say, well, maybe your carotid has a little bit of a kink in it, like that. And say you ate an overly rich diet, and so a little crud starts to build up in there, and it starts to block it off. And so you can. That's the kind of stuff you can. You didn't eat enough broccoli, and you can see stuff like that. So, how do you visualize the blood vessels? Well, it turns out there's an easy way to do it, which relies on the fact that if you're imaging something like the brain or some blood vessels in your neck, the spins in there are moving. And this is a very sort of strange thing to think about, sort of just physically. How does this actually work? So, but say you just do like a regular gradient echo localizer scan. It's a 1 minute, 10-second scan to just find out where a couple slices through the brain to find out where it is so you can set up your longer scans. If you look at a scan like that, you'll see the carotid is white. Very big signal coming out of the carotid. And so why is the carotid white? It's because you're doing some imaging in the head, but there's some spins, and those imaging, those protons in the head are getting hit by RF because it's a frequency selective pulse. And so those guys are getting saturated, meaning that they're losing their longitudinal magnetization from getting hit by all the RF pulses. And so there's not going to be as good a signal. Whereas if you've got some protons in your heart, and your heart like pumps them up, those guys are going to be fully magnetized, MZ0. They're going to have sort of the full magnetization. And so when they come up into the brain, they'll have more longitudinal magnetization to work with. But think about what's actually going on. It's kind of amazing. Like the protons in your heart are not just sitting there; they're just bouncing around in the blood vessel. They're inside a blood vessel, and a blood cell, and the blood cell is like sort of waffling around as it gets pumped through the heart. But the protons are like little ball bearings inside those macromolecules and inside those water molecules, and they're polarized, meaning there's more of them sort of like pointing up at 54 degrees away from, whatever the B0 direction is, than there are sort of like, that's what longitudinal magnetization is. It's a excess of ones that are sort of lined up with the field versus the ones that are not lined up with the field. Not a big excess, 1 in 10,000, something like that. But if you actually think about that, they're lined up in spite of the fact that the molecule that they're in is bouncing around. So the molecule at the end is like bouncing off the walls like that, but they're just sitting there like, I'm lined up. And so then it goes through, and those molecules just bounce up, and then they'll flow into the head, and they'll have extra longitudinal magnetization. And so then when you do an RF pulse, you get a nice big signal out of them. So that's basically the principle of the time of flight, TOF. Time of flight is what it means. It just sort of doesn't really make sense, time of flight. But basically, what is it? It's just basically the inflow of unsaturated, which means big longitudinal magnetization spins. And what can you do to sort of increase this? You can pre-saturate the region. Saturate region of interest. And you can do this on the scanner. You can set up a saturation band. So like saturation, that just sort of seems like saturated fat. What does that mean? It just means do a 90-degree pulse so that you turn all the longitudinal into transverse, and then let the transverse decay away so that there's no signal from it. So basically, what you can do is that you've got some blood vessels, coming up into the brain here. So here's the brain, and so if you want to see these blood vessels, what do? There's the heart down there. Okay, heart. So what you do is let's blast off all this. So saturate that whole mess. So the brain, so any of the blood that's coming, back down, because the brain goes, the blood, like Aristotle saw, brain goes up, blood goes up to the brain, and then it comes back down. So let's saturate all the blood that's in the brain, plus all the blood that's in the blood vessels we want to look at. And then, so that blots out that signal. Then wait a little while for some unsaturated spins to flow in. So wait until they flow in just a little bit. You don't have to wait very long, And then do an image. And so what you should get is just blood vessels, and that's pretty much what you can see. So how do you do it? Well, it's sort of it's sort of strangely just like every other pulse sequence. So like do a 90, but do that 90, do a 90 up here. And then, make an image. So give a little delay to decay MT. So we made some MT from that, and that would make a huge signal. So let it decay, then make image. And what you'll see is just basically blood vessels. It just looks like these white blood vessels. Now, if you've ever looked on the scanner, what do they do with that image? You can immediately turn it into like a rotating 3D picture of these blood vessels, and you can look for little crud, see if there's any crud built up there without doing any X-rays. And so how do they make that image? It's a very simple kind of algorithm for making that 3D image. At first, you would think like there would be like some high-powered computation to reconstruct that image in 3D so that you could actually move it. But all you got to do is you just have a block. So you just recorded a block of data with some blood vessels in it. So here's some, there's some blood vessels in there like that. And so then what do? You just take that block of data and then basically just make lines through it in one direction. So there's the block. So just start somewhere, like start here along the back face, and just make a line that comes out to the same point on the front face, and then just take the max. It's called a maximum intensity projection. And so the projection is we just go through a line of pixels and just find the brightest one. That's it. So if you do that, and then just do the same thing, for all these things, you end up with a 2D image. So what is that? That gives you a 2D image, single 2D image, and you'll see some blood vessels in it. And so there's your picture of blood vessels. And then what do? Just do it again. So this is a very easy computation because you just go through the block, finding the maximum. And so now go through the block again, but start off at this point and then come across. So instead of going to the same point, come across to like that. So basically just go through the block in a different direction. I over-exaggerate. You could just go through the block 1 degree away from the last time you went through the block. And then what do you do with that? You just make that into a picture. And then you'll get a picture that basically has the blood vessels rotated in it. So now you'll see the same thing, but they'll be rotated around a little bit. They'll look at a different angle. So if you just do that all the way 360, you can very quickly get a little rotating picture, ghostly rotating picture of just their blood vessels, of all their blood vessels coming up into the brain. So it's a very convincing thing to look at. So that's a time of flight. And so why did I mention this in the context of magnetization transfer? Because you can use magnetization transfer to improve the contrast. And so how does that work? Well, so if you do a time of flight, so basically just, saturate this block, wait a little bit, and then take an image of it to let the spins come into it. So you do that image, but then to increase, also do, add a magnetization transfer preparation to suppress, because remember, the idea was the voxels where there was some transfer from these proteins, from these excited transverse magnetization of proteins, it ends up sort of leaking over to the water signal, which is our main signal, and lowering it and suppressing it. And so to, suppress macromolecules, and that would be like, where are there a lot of macromolecules, like, in tissue, brain tissue. So we don't want to see that. So all we want to see is the blood vessels. And so if you turn on a magnetization transfer pulse, like some of these things were just kind of like figured out empirically. Well, it looks better. I wonder why. So if you turn on a magnetization transfer pulse, what will happen is it will suppress the background, and you'll just see the blood vessels even clearer. And so that's commonly done for a time of flight scan. So you can see it's kind of, a lot of these things are kind of like strange little hacks that you can do, but the end result is you can do a completely non-invasive imaging of everybody's vessels without having to inject any toxic contrast agent or use X-rays, which are kind of damaging to tissue. And so it's a very useful way to sort of quickly visualize, just in a couple minutes, visualize the blood vessels. So that was magnetization transfer, relevance to time of flight, how time of flight works. And we'll talk more about time of flight-like scans when we talk about perfusion or arterial spin labeling. And so in this case, what we're doing is we're just letting some spins that weren't saturated flow into our slice to give us some contrast. But instead, what we could do is we could actually excite the spins, like invert the spins, like flip the spins to, a minus mz somewhere, like say down in the carotid, and then wait for those inverted spins to flow up into the brain as they recover. And so we could specifically label some spins on purpose instead of just sort of relying on the fact that they're not saturated down there. That's how a perfusion scan works. And so we'll talk in more detail about that as kind of like a more quantitative method of trying to measure the amount of flow coming into tissue, as opposed to just seeing the blood vessels. And with perfusion imaging, we're essentially trying to measure like how much blood flow went into the brain, as opposed to just seeing a picture of the blood vessels. So we'll come back and talk about that later on, after we talk about diffusion. And one of the topics, I already went over this once. It's quite a confusing thing, so I forgot to put up some of the labels last time. So I just want to talk about how to visualize off-resonance excitation. So the way we visualize on-resonance excitation is very straightforward. We just imagine we're in the rotating coordinate system, and then we just do a flip. So how we just have a 2D coordinate system. It's just got m longitudinal and m t going this way, and then we say, well, we've got some magnetization longitudinal, and then we just do a flip down to here. We don't think about any precession. We just think about those stationary vectors that are just being rotated. So what are we doing? We're actually sort of thinking in the rotating coordinate system. And so let's just go over how we would think about an off-resonance pulse in the rotating coordinate system. So to start off, we have the laboratory frame. So this is what's actually there. And so what is actually there? We've got our coordinate system, and then we have a magnetization vector. That's whatever the local magnetization vector is. And then we've got the B, So this is our M vector, and this guy is the B vector. And our equation was, block equation was the change in the magnetization vector for a small amount of time. That guy's a vector, just equals the, whatever the magnetization vector currently was, cross-producted with whatever the B vector was. And when we work through that equation for a stationary B vector, what do we get? We get like, this precession like that. And what is the dm dt vector? It's a vector that's perpendicular to both of these. It's perpendicular to both the M and the B. So that guy goes there. And that tells us, what the next time point is. But that's just a stationary B, and what's going on when we actually do an excitation? Well, we're adding another rotating. So here's our rotating B1. So we're adding. So that's our B1. So our B1 is rotating around, that guy right there, and that's added on to this guy. And so now we've got a more complicated situation. So what's going on is the B is kind of wiggling rapidly at some frequency, might not necessarily be the same frequency as the M. So the M is going around, and then at the same time the B is wiggling, possibly at a different frequency. So you can see what the problem is. It's just kind of hard to sort of wrap your head around what is the effect of that on this equation. It's going to be like kind of a mess of a complicated combination of things. And so one way of simplifying it is to consider the rotating frame. So the rotating frame is what we're already doing when we think of it this way. And so the rotating frame is, let's just imagine that we're spinning around at the same speed as the precession frequency. So if we spin around at the same speed as the precession frequency, it's going to basically stop the M from rotating. So the M will just be stopped in its tracks. So it'll still be tilted, but it will be stopped in its tracks. And so assume that we hit the right frequency. So if you hit the right frequency, it just stops this. And what's the implication of that? So the implication of that is that, so M vector stops. It's stationary, wherever it was. But what does that imply? That implies B0 equals 0. Because if B0 equals 0, when we do the cross product with 0, that's going to give us 0. So, the cross product of any old vector with a 0 is 0. So that means there's no update. The dm dt will be 0, so it doesn't go anywhere. So that's all good. But one big advantage of looking at it this way, and the reason why everybody sort of thinks of it this way, is that we can add the B1 into here. So say we were on resonance. So here's our M. And so what we can do is, add on resonance B1. So the B1, if we're doing the right frequency that we were already going at, so if we're doing the resonant frequency we're already going at, that B1, it will have a particular phase, but let's just assume that it's this it's this particular phase. So there's the x-axis. So here's our m stopped because we are rotating around the same speed as the m, and here's the b1. It's actually just the b because the b0 is 0. So here's the b1 vector, also stopped. So so this is b1 vector also stopped. But now we can still operate that equation. That's the beauty of it. So we can still operate our standard equation on this situation with a stopped b1 vector. And so how does that work? Well, you've got the b1 vector and you got the m vector. You take the cross product that's perpendicular to the plane made by the b and the m vector, and then it's a small vector that tells you which way the m vector is going to go. So this still works. So this still works. And so that's why when we're constantly saying we're doing a 90-degree flip, we're thinking in the rotating coordinate system. And what's causing the flip? The b1 is causing the flip. And so what is the direction that it's going to cause the flip here? Well, it's going to make a dm dt that looks like that. And so it will cause basically the m vector to precess around the b1 vector. So that's the bottom line. So m precesses very slowly compared to like the 123 million, precesses around b1. And so that movement is the same kind of precession that we have over here. It's just much slower. So it takes a millisecond or half a millisecond instead of a hundredth of a microsecond. So, but it's the same exact equation. And so and then that also allows us to consider like different phases, because what is the different phase of the b1? That would be somewhere else. Somewhere else in the xy plane. So here's the xy plane around like that. And what if we had a b1 pointing in that direction? It has a slightly different phase angle. Here's the phase angle of that one, but here's so let's do it the other way around. So here's the phase angle of this guy, but here's a different phase angle of this guy that comes around like that. And so in that case, what would happen is you would have a different computation, and instead of like aiming down toward the y-axis, if you put that different phase of the b1 oscillation, you would rotate in a different direction than straight down to the y-axis. And if we go back to again, the value of looking at this, what is the different phase? It just means like that very rapid wiggling that the b1 has added to the b0 is like a quarter cycle ahead. It's like a quarter cycle ahead of, say, where this current m vector is. And so we can sort of visualize that complicated situation much more simply by just sort of considering the stopped b1 vector causing the stopped m vector to slowly precess around it. Okay, so that's where we already were. And now we're ready to do off-resonance. So how do we think about off-resonance? So here's the off-resonance. So now we want to look at, we'll stick in the rotating coordinate system appropriate for the b1 vector, but we'll assume that the m vector is precessing at a different rate than our rotating coordinate system. So we'll look at it from the point of view of the b1 vector. You can do it other ways, but let's just do it. Let's do it that way. And so now what's going to happen? So now our rotating coordinate system is slightly off for the m vector. So here's the m vector, same one. but the rotating coordinate system is slightly off for that m vector. And so that m vector is going to start to precess. So it's going to, not very fast, but it's going to be precessing like this, slowly precessing around like that. And what does that correspond to? You can think of that. So here's our m vector. You can think of that as basically being due to a b zero. So here's like a fake b0, which is the amount of b0 that you need to make that thing precess. What is that fake b0? It's just the difference. It's basically equivalent to the difference between the precession frequency of the m and whatever the precession frequency of your rotating coordinate system is. If you're off a little bit, then it's sort of like a pseudo b. It could be negative also, depending on whether you're above, whether your rotating coordinate system is above or below. But it sort of like adds, so we'll call this, apparent B0. So it looks like a B0. But now we're going to do RF excitation. So here's an actual B1, and we've set the rotating coordinate system to the same frequency as the B1, so the B1 is stopped. So this is the actual B1 here, actual B1 vector, but stopped because we're rotating at the same speed as it. And so what is the result? Result is we add these two guys together and we get this vector. And so what is that vector? That's the B the total B effective vector. Effective. So that's this guy right here. So now how do we view what that does to the m vector? It just causes the m vector to precess around that B effective. So now, if in this case, what happened is, if the m vector was in the zy plane, when we turned on the B1 vector, it would cause it to aim right toward the y-axis. But now, in this case, what it's going to do is this guy is going to sort of come out at you like this. It'll come out at you and sort of precess around something like that. So it's going to be precessing around in a different plane. It won't be precessing around in the zy plane. It'll be precessing around in a plane that's like tilted out at you. But it's sort of understandable. So we can sort of predict where it's going to go. And what does that mean? We're going to predict, what angle, what is the flip angle going to go to, and we're going to predict what the phase is. What is the actual sort of phase of the oscillation? So that's, when we get back to arterial spin labeling, we'll see that by slowly sweeping an RF pulse through the center frequency, you can do an inversion. And that's relevant to how arterial spin labeling works. So you can sort of imagine if we start off above or below the frequency and then sweep through the center frequency, we can sort of create like a positive fake B0, and then going down to 0 and then making a negative B0. And so you can sort of think of it as a way to invert spins. So we'll come back to that and just go over it a third time. And it took me a long time to sort of try to figure out like what I saw this picture repeatedly, and the first time I saw it, I was just like, ooh, I just didn't process it. But I finally got it, going through it a couple times. And so it's just basically a way of trying to visualize what's going on that's easier to sort of think about than the actual situation, which is you've got precessing protons precessing very rapidly, and then you've got a B field that has a B1 field added to it that's wiggling possibly at a different frequency versus these, and what's the end result of applying this equation, repeatedly. It's hard to sort of visualize. This makes it a little easier. You can sort of think of it as this is effectively where the B field is. Okay, before I go over, Stan, we'll stop recording there and take some questions. Okay.